So in this tutorial, I'll try to have more like a recap from a high level, like a very brief overview of what native mass spectrometry can do, can help us understand the protein structure and function, and also what capabilities we're offering at EMSL. So it looks like it's about time, so I'll just start. And the beginning especially for those of you who are not very familiar with native mass spectrometry, I want to take a step back just to explain. Oh, my slide is not advancing. Oh, okay. So just like give like a very brief overview of what is native mass spectrometry and what is different from the typical proteomics experiments we all are familiar with. So as we are, some of you are very familiar with, is that most of the proteomics experiments being performed by mass spectrometry has been very peptide-centric. So we started with proteins. Let's see if I can have my pointer. So we started with intact proteins or native proteins extracted from biological matrices, and we digest them in the peptide using some specific enzymes. And then we use liquid chromatography or some other separation techniques to separate those peptides in different retention time. And then we can use mass spectrometers to measure the peptide and also fragment the peptides to identify what sequences they are. And with informatics tools, we can automatically identify what peptides are in the sample, and this helps us understand what proteins are in the sample. And this analysis can be also performed quantitatively to help us understand the relative abundance of proteins in the sample. So those analyses are based on the peptide because we're inferring the presence and also the abundance of proteins based on the peptide data we measure. And this is being referred to as bottom-up proteomics. So the bottom-up proteomics is very robust and is very good for both qualitative and quantitative analysis. And we know a lot of people are working with EMSL using this proteomics capability, but there are some limitations. So for example, if you have your protein and it's been post-translationally modified at several different locations, so you may have different combinations of these modifications on different forms of this protein, as you remember from Professor Albert Hecht's talk this morning. It's like there are different proteoforms that are potentially having different functions in cells. And when we use the bottom-up analysis, we chew up the proteins into smaller peptides, and then we lose this connectivity between the different combinations of the post-translational modifications or mutations or terminal truncation of proteins. So if we study the protein at the intact level, this means like we're having a top-down analysis of the proteins. This gives us the benefit of seeing the big picture of the combination of different modifications and help us really define the functional proteoforms in cells. And this is a nice representation from Professor Neil Carter's group showing if you are doing bottom-up proteomics, we're getting very nice measurement on those pieces, jigsaw puzzle pieces. But we really need this top-down analysis from the intact level to help us define the complete picture of the protein. And we also know that the living proteins in cells, they have higher order structure. It's not just the primary structure that is important for the protein function. And unfortunately, most of the current proteomics pipelines at EMSL focuses only on the primary structure analysis. As you have heard from Professor Jim Bruce's talk this morning, we can use different, such as cross-linking techniques to help us understand high order structure, even with the peptide-centric methods. But also from the other talks, you have seen that we're using native mass spectrometry, you can even probe the high order structure of the proteins. And just to show you a very basic example, and you probably remember that from the talks in this morning, like we do use the same electrospray ionization for this native protein analysis. The major difference is that we try to keep this protein in non-denaturing conditions. So normally if we do like LC-MS or like peptide analysis, we like to use very volatile solvent, like organic solvents, including like acetonitrile or some formic acid or some other acids to help ionize, protonate the proteins to improve the signal. And in this case, because the conditions are very denaturing to the proteins, we're only seeing the monomers of the protein with a lot of charge, indicating that they're unfolded. But if we use a neutral solution, such as ammonium acetate at 100 millimolar or higher, we have this very different spectrum indicating that we're detecting the intact pentameric species, which are non-covalently held together. I consider they are very different also in the native mass spectrometry, because we are measuring those intact complexes. They are also very much higher mass than when you normally do the denaturing analysis. So that was in a nutshell what native mass spectrometry is. And the next I'm going to show the highlights from some publications in the native mass spectrometry field to show what native mass spectrometry can do to help us understand protein structure and function. The first thing I want to say is the mass measurement is very useful for determining stoichiometry of protein complexes. And you can do like different protein-protein complexes or protein-ligand complexes, protein-metal complexes, or protein-nucleotide complexes. And this is a quick example showing how you can look at the sulfur binding between the two proteins. And you can easily tell the binding of the sulfur based on the mass shift in the mass spectrum collected under non-denaturing conditions. And the two examples on the right are actually from Professor Abraham's group. And the first one is looking at the virus particles, and the other is look at ribosome RNA complexes. The other thing I want to expand a little bit is the protein-ligand interaction measured by native mass spectrometry. And the benefit of using mass spectrometry is that this can actually be done in high throughput to screen ligands against target protein. So this is a manuscript published from Ron Queen's group at Griffith University in Australia. And they were doing this drug library screening using native mass spectrometry. The concept is that when you have a target protein, you can mix it with all kinds of different ligands, and you can easily see if it binds to the protein based on the mass shift to the protein. And you can quantify the binding strength based on the relative abundance of the apoprotein and the holoprotein. And so you can see some of them are not really binding strongly. And you can do this in a very high throughput manner to fast screen the ligand. Even if you don't know exactly what the ligand are based on the mass, you can tell what the identity might be. And this can be helpful for screening drug libraries, such as like natural products. So this example, you remember, I think Joe showed this morning, that the native mass spectrometry can be also useful to look at the protein dynamics. And this is like circadian clock complex in cyanobacteria. The concept is that this protein in the day and night cycle, it changes dynamically. And using native mass spectrometry, you can easily see that the complex will assemble and disassemble in real time. And using this information, you can optimize the condition to perform cryo-EM analysis and get high resolution structure of all the different intermediates in this process. And also, you have heard from the speakers this morning that native mass spectrometry can be also used to study membrane protein complexes. So normally we would imagine detergent is bad for mass spectrometry, but actually it's good for keeping the membrane proteins intact, and we can then detect those intact complexes in the mass spectrometer. And there's a nice, more like mini review article from Professor Michael Marty, summarizing the different membrane micelles or membrane like environments like nanodiscs that can be used in native mass spectrometry to like stabilize membrane proteins. And the concept is that once you have this membrane embedded in nanodisk or micelles, you can electrospray them into the gas phase, and then you can activate the proteins and release the membrane proteins from the detergent. And in this way, you can get a very high resolution measurement on the membrane proteins. There was a very impressive publication recently from Professor Karen Robinson's group showing how they can actually isolate native mitochondrial membrane, the intact membrane, and then spray into the mass spectrometer and then activate and release those native membrane protein complexes and measure them with mass spectrometer. And this is truly impressive because you can actually look at those native complexes endogenously from biological matrices. So it's more than just the math that this such experiment can be performed. If you remember Varun's talk this morning, there's also this ion mobility dimension we can add to the native mass spectrometry analysis. So he did a nice introduction this morning, so I'm just trying to quickly summarize it. So ion mobility is orthogonal to mass spectrometry, and it's a gas phase separation technique that is analogous to electrophoresis. So you do have like an electric field that's pushing the ions across this drip tube, and there are different designs for how the drip tube can be used. But in general, you have the electric field pushing the ions, and then you have the another force from the collision of the protein ions with the base gas. And this will basically slow down the ions based on how many collisions they will have with the base gas. And this separates the proteins based on their charge. The faster, the higher charge will be pushed faster. And also the more collision you have because of the larger surface area of the protein ions, it will be slowed down more. And in this way, you can separate the ions based on their different drip times. Usually they are on the order of milliseconds or much longer, depending on the design of your drip tube. And based on this drift time measurement, you can plug into the equation, and then you can calculate the collisional cross-section of the ion, which is a measure of the size and shape of the protein ions from this experimental data. So the quick example I'm showing you is also kind of the same concept of what Varun showed this morning. So the one big benefit of biomobility is that you can actually separate a lot of the species that may not be well resolved in the mass spectrometry analysis alone. So this graph shows the mass to charge axis from the mass measurement in the x-axis, and the y-axis shows the drip time of the ion mobility separation. And the color basically represents the relative abundance of the species. And as you can see here, those species, there are different species assigned to different structure of the complexes between the two proteins. And they have all different kinds of combinations. It's a very heterogeneous mixture. And if you don't have the additional separation from the ion mobility, a lot of the peaks will overlap in the mass axis, and you may not be able to resolve them very well. And with this ion mobility separation, you can easily define those heterogeneous mixtures based on their mass. And this helps us to build models. And this eventually will help us explain the ultracentrifugation data a lot easier, because we have this species of the whole in the whole assemble. So it helps us to feed the data and understand how those two proteins interact and assemble in this mixture. The other factor that from the ion mobility that are very beneficial is look at the conformational change of proteins. In this example, I'm showing the proteins without the ligand on the top and the plus ligand at the bottom. So the graphs are basically representing the collisional cross-section distribution measured by the experiment. You can see how the collisional cross-section distribution was broader when you don't have the ligand. You have almost like a second distribution up here. But once you bind the protein to the ligand, it seems to remove that distribution, indicating that the proteins are maybe less flexible and more structured when you bind to the ligand. This can be helpful to study the like allosteric effect of ligand binding of proteins. And of course, you can also just study protein structure in this way. If you have the collisional cross-section measurement, you can also build computational models and try to fit it to the experimental data and determine which structure is more probable. The other stuff I do want to show for ion mobility is also covered by Varun's talk, is the collision-induced unfolding, which means that you are doing thermal melt experiment in the gas phase via collision with the gas molecules. And as you start, you can control the collision energy very well. Basically, you are slowly heating up the protein, and this protein starts to unfold, and you can monitor the change of the conformation based on the change of the drip time you measure in the ion mobility experiment. And this can be useful to tell structural differences in proteins, as you have already seen this morning. And this is just another example showing how you can tell the slight differences in disulfide binding in the IgG molecule. So they have both of the molecules have almost the same overall shape, the starting point of the drip times. But if you heat up slowly, the drip time distribution changes, and then you start to see some differences here as it's been unfolded. And this can be very useful to reveal structural differences in proteins. And I believe you'll probably be hearing more of that tomorrow in tomorrow's sessions. So those are all great stuff to see what native mass spectrometry can help us getting structure information. So so what's behind the scenes? So what are the instrumentation we need to do the native mass spectrometry analysis? So as you imagine, if the experiments are so great, so why like still feels a little bit difficult to do those experiments? It's because like you typically need some specialized instrumentation to perform this experiment because of the high mass of those native protein ions. So a lot of the commonly used proteomics instruments are usually restricted to relatively low mass to charge range, so you won't be able to detect those or transfer them effectively for analysis. And there are several different models that are compatible with native mass spectrometry analysis. And at EMSL, we have almost all of them except the Bruker team stuff. And most of the native mass spectrometry experiments we're doing are on the thermal UHMR Orbitrap and also the water CNAP ion mobility time-of-flight instrument. And you've heard people using both of those instruments from this morning's talks. And also some people have used Agilent IMS as well. So the mass resolution is important, and it can be very useful for especially to look at small mass differences on large protein complexes. And each of the platform has different advantages and disadvantages. The options for ion activation is actually extremely important for native mass spectrometry, which I'll discuss a lot more because it's critical for dissociation and dissection of protein complexes. And the options for ion activation can be limiting factor depending on which platform you're using. But there are a lot of efforts trying to commercialize and make this workflow more streamlined. And I'll be showing a little bit more detail in the next few slides. So why ion activation methods are so important for native mass spectrometry? Because if you are spraying the proteins under non-denaturing conditions, they like to stick to a lot of salt or solvent molecules, and it basically broadens your peak and it destroys your mass resolution. So you need some activation to remove those non-specific adducts and improve your mass analysis. But at the same time, you don't want to over-activate your protein, and then it will basically destroy the non-covalent interactions. So it's a fine balance you have to tune depending on different proteins. And the other thing is that for native membrane proteins, especially because as I've just shown, like you have to use some kind of detergent environment to solubilize and protect the membrane protein complexes. But to do the mass measurement, you have to release the membrane protein complexes for mass detection. And you need specific ion activation parameters to optimize the whole process without destroying the membrane protein complexes. The other things we can make use of for ion activation, just like we're doing the normal peptide-based proteomics experiment, we would benefit from fragmenting the backbone of the proteins because they will give unique peptide fragments to help us identify what proteins are in there in the complex. This is very, very useful if you're working on like unknown protein complexes. And also, there are different methods you can use to dissociate the non-covalent interactions while keeping like in a very well-controlled way, so you can actually map the subunit connectivity of the protein complexes, which I'll have an example to show you later on. And all those activation methods can be useful for probing high-order structures or proteins. So how to actually perform those ion activation methods? So the collisional activation is very commonly used, especially the collision-induced dissociation. Especially you are colliding your protein ions with gas, like usually nitrogen or argon. This is almost available in every commercial mass spectrometer right now, used for proteomics experiment. There's also a way to collide the protein with surface target. It's called surface-induced dissociation, which I will be showing you more examples later. This method can heat up the proteins faster and resulting in dissociation with minimum unfolding to help us map subunit connectivity. This technique is currently not widely available yet, but there are new instruments that have been announced having this capability. And we do have this at EMSL. So for electron-based activation, you're interacting your protein ion with electron beam, either with very low energy or higher energy, depending on which method you are using. And you can also transfer electrons from radical ions. And all those, we just call them as EXE, basically electron-based activation. And it's commercialized in some instrument, but typically you still require some modification. There is a company called Emission who has commercialized this device as like add-on to other commercial mass spec platforms. And there's also a photo activation, which you are shooting the laser, either ultraviolet or either ultraviolet wavelength or infrared wavelengths to the protein, and as the protein absorbs the photons, you will be activated. And for the collisional-based activation for protein complexes, it's very effective at disrupting the non-covalent interactions. For electron activation, it's very good at cleaving the backbone of proteins without significantly disrupting the non-covalent interactions. For photoactivation, you tend to get the mixture of both, depending on what wavelengths you are using in this experiment. And as I was saying that a lot of those techniques, except the CID, are not as widely commercially available. So you typically need specialized instrument and operation. But the things are changing fast because the companies are making a lot of efforts trying to make this more easy to use and commercialize, so everyone can start to use this to study the protein structure. So with all those nice activation techniques, all these options, this is more like a very inspiring example from Zhou, that you can actually identify unknown bound ligand. If you start from a native protein complex, membrane protein complex, you can start to activate and dissociate the little pieces from the complex and dissect them to identify what they are. And if you do this kind of analysis in the proteome at a proteome scale, and this has already been demonstrated by Professor Neil Keller's group and also Liang Sun's group, that if you do a front-end separation, you can simplify the data, simplify the samples, and then you can do this kind of a top-down analysis on the protein complexes to do this native proteomics experiment. This is still at an early stage of development, but as you can imagine, if we can do this effectively at the proteome level, this would really help us get a lot of extra information on the proteins in biological samples. So not just only the protein abundance, but a lot of other structure information as well. So a quick recap on this part. So we have all those different activation techniques that will help us to access different levels of protein structure information. As I was saying, the activation method will dissect the protein at a different structural level. And usually you will get complementary structure information by using different techniques or a combination of different techniques. And currently, the photo dissociation, electron-based dissociation, and surface-induced dissociation are either restricted to specialized instruments or require some custom modification. And we just had a recent prospective article to cover this this topic of the recent advances on top-down analysis of native protein complex to help us understand a high-order structure. And it's just accepted in chemical science. And if you're interested, they should appear online pretty soon. So the recap for this part of the talk. So I hope I've shown that native mass spectrometry can be very useful for high order structure analysis. And I would want to say that the analysis is usually pretty fast. It takes probably only minutes for most of the routine samples. For some more challenging samples, it will take longer. And the nice thing about the mass spectrometry analysis is high versatility, because we can analyze soluble or membrane protein complex or even intrinsically disordered proteins. There are some very nice publications on that too, which I don't have time to cover. And as you have, you probably remember from this morning's talks, there's a very big highlight on how native mass spectrometry or other mass spectrometry techniques can really deep into the heterogeneity of the proteins, either as like purified complex or simply, or even in cell or in vivo. And it's very complementary to other structural biology tools such as cryo-EM, crystallography, or NMR. And I do have a few examples to show later. Or you can also you also probably remember from the other talks today, and also the past few days discussing the complementarity between different techniques. So there are still some downsides to the current technology for native mass spec. So most of the work has been performed into on like a relatively pure samples. A lot of them are like structural biology projects where you have recombinant proteins, and you have like remember from Albert's talk this morning, you can extract the native proteins from endogenous matrices and do such analysis. But it's still quite challenging to do this at a whole proteome level. That what people have been doing for bottom-up proteomics. So there are still a lot of development required to have this front-end separation to simplify this and also improve the mass spec detection and speed to really study the native proteins at the proteome level. And the other thing is becoming less of a problem right now. It used to be only restricted to some academic labs to do this kind of analysis because it did require specialized instrumentation and operation. But now, because of the commercialization and the great efforts by the mass spectrometry vendors, a lot of the processes have been streamlined. It's almost like a plug and play. You once have the sample loaded, you can just get the data right away. But still, for the more advanced activation techniques, it's not completely widely accessible yet, and there are the developments on the way, and I'm pretty sure they will be accessible in the near future. So I will be showing a quick video on what a normal, like a very abbreviated process of what we typically do for native mass spectrometry analysis. So native mass spectrometry is a relatively new technology and does require specialized instrumentation and also operation. So right now we're offering this technology to relatively pure protein samples, and we have been working with the cryo-EM group to offer this new technology for users who are interested in structural biology problems. And we're still actively developing new technology under the EMSL internal project to improve this and make it more high throughput and more accessible to other users. So we now offer this native mass spectrometry analysis to purified proteins. It can be either recombinant or purified from biological tissues. And when we receive the sample, we will do the buffer exchange to make it more compatible with mass spectrometry. But for storage, when you send the samples, I would recommend keeping the protein in the buffer. That's best for the stability. To exchange the buffer, we use this kind of size exclusion columns, spin columns, to exchange the buffer into ammonium acetate solution, which is compatible with mass spectrometry. To start, we will actually first condition the column with ammonium acetate solution. And we'll just use a benchtop centrifuge. And we prefer to use the refrigerated centrifuge just to keep the protein stable. And we will repeat this for usually 4 to 5 times to maximize the exchange efficiency. And once the columns are conditioned, we will load the samples onto the column, and all the salt and the other small molecules will be trapped in the size exclusion column, and we'll basically get the protein solution directly in the final centrifuge step. So we make those nano electrospray capillaries to electrospray our proteins for native mass spectrometry experiments. And we make most of our capillaries in-house. So we have those glass capillaries, and we'll use this glass puller to make those tapered capillaries to do the experiment. So to do this, we first have to position the capillary into the puller arms. And once it's in position, we'll start the pulling by heating up the filament inside. And so, as you will see just in a few seconds, the filament will heat up and will slowly pull the capillaries apart, and that will form a very nice taper tip at the beginning of the capillary. So that's finished, and we can take the capillary out and save it in boxes, and we'll use that for later use. If you want, you can also check the size of the capillary under a microscope or electron microscope. So to start the experiment, we first have to load the sample into the glass capillaries we made. And we'll have to trim the capillary if it's too long to fit on the instrument. And once it's the right size, we use this long gel loading tips to load the samples into there. And this is our buffer exchange protein solution. And we usually take a few microliters, and we try to get rid of the bubbles in there. And then load on this stage. And this is basically a platinum wire for conductivity of the electrospray voltage. And we put it in there so the wire is actually in contact with the protein solution. We fix this sprayer on the stage, and we slide it in so that it's very close to the inlet of the mass spectrometer. So now once we start the electrospray, we'll see the protein signals coming out in this mass range. This protein is C-reactive protein, and it's a pentamer of 115 kilodalton. And because we're measuring mass through charge, we actually see a few peaks that correspond to different charge states of the same protein. And what's interesting here is that we can actually isolate the protein, the pentamer protein, so we can mass isolate a species in the mass spectrometer for this protein complex, and then activate, use collision-induced dissociation in the instrument. And this pentamer will be activated and it will fall apart, releasing the monomer of the proteins. And this experiment, we can actually get some information on the composition of the protein complex. And we also have a lot of other techniques we can use to get further structural information of those protein complexes. So we also have the function to have ion mobility separation in parallel to the mass spectrometry detection. So I just activated the ion mobility mode for this instrument, and now we have two windows. So we can not only just analyze the proteins by the different mass charge, but we can also actually separate the proteins in the gas phase based on their shape. And that gives us this extra dimension of the drip time separation for this analysis. So after collecting the data, we can either manually look at the spectrum saved, or we can use software tools to help us. So we have this, we use this Unideck academic software from University of Arizona, written by Professor Michael Marty, and it helps us to automatically convert the raw data into a mass distribution, as seen here. And this really helps us to speed up the analysis. For the ion mobility data, we can also load the data in the StripScope software, so we can actually see the two-dimensional spectrum with the mass to charge on the x-axis offered by the mass spectrometry measurement, and also the drip time separation offered by the ion mobility. And we see those peaks corresponding to the CRP protein we just analyzed. But we're always looking for new challenges for scientific questions, and I would be happy to talk to you if you are interested in this new technology. And thank you for watching, and I hope I can collaborate with you very soon. Okay, so that was a very abbreviated process of what we normally do here. And usually for unknown samples, we'll have to optimize a lot of the conditions to make sure it's compatible. And so for the sample preparation protocols I was just showing, that before we run the mass spectrometry analysis, we need to buffer exchange the samples into volatile salt solution for native mass spectrometry. And we have options for ammonium acetate, formate, or bicarbonate, depending on what pH your protein is stable in. And we use this size exclusion spin column or the membrane filter based on the size of the molecule as well to remove the low molecular weight components, usually the salt or some other contaminants. And we typically work on micromolar protein concentration and on the order of tens of microliter, and which transfers to kind of microgram of protein. And this is kind of the minimum requirement. If you have more sample, it's usually better because we can optimize the condition and try a few different attempts to with different types of experiment. The optimum size range for the current instrumentation workflow here for the native proteins is megadalton or smaller. The bigger proteins, they're harder to do because of the size. But it's, as you have seen today, like Albert showed quite a lot of impressive data going much higher. So it's also possible, but right now the most common workflow we work on usually hundreds of kilodalton for the protein size. And for data analysis, I've just shown that we do a lot of manual analysis to just really confirm we have the correct assignment. But also there are software tools available to us, especially Unideck is open and free to everyone. And you can use it to easily transfer your raw data to mass distribution. And there are a lot of other modules in there to help you process the data. Because we also have the intact mass from Proteometrics at EMSL. It's like a paid software license, and it will also help us to deconvolute the raw data into intact mass distributions. So if you are interested in sending samples for native mass spectrometry analysis at EMSL, so there are several things you need to consider. So regarding the sample storage, and there are a lot of commonalities between the native mass spectrometry analysis and the cryo-EM. If you remember from Trevor's tutorial on Monday, we want to minimize, generally we want to minimize any other components in the solution. And the standard salt buffer is usually fine, and we can easily remove them with the buffer exchange column. The detergent, if it's non-ionic, and if you're working on membrane proteins, and those are necessary to stabilize and solubilize the protein, that's fine. But if you don't need the detergent, I would recommend not to add any detergent, because if it's concentrated enough, you have those micelles, and they are actually a bigger size in the same range of the proteins, and they will be harder to remove. And try to minimize concentration for any other stabilizers. So some proteins, they may require some cofactors or some metal ions to stabilize itself. So we can deal with relatively low concentration, like maybe like up to millimolar, but like not anything higher, and it will actually cause some problem if you have so much of the other non-volatile stabilizers in the solution. And for things to avoid, ionic detergents are very difficult to deal with because they are easily ionized and it will mask your signal of your proteins. Synthetic polymers, because of the size, it's harder to remove them using the size separation we typically use in the sample preparation step. And the glycerol is okay, I think you can remove it, but if you use a lot to stabilize it, it's in the past, I've had a bad experience with it. So, but ideally, just try to minimize the components in your sample, and that would really simplify the analysis and improve the quality of the data. So the second half, I'll be just showing two quick case studies from recent EMSL project. I want to have a short pause here if anyone has a question regarding the whole workflow and if there's anything you are unsure about for what native mass spectrometry can do. So Beth, could you help check if there's anything coming up yet?
I don't see any questions yet, but everyone, you're welcome to unmute yourselves and just ask your question directly, or you can post it into the chat window.
Oh, okay. I'll just give a minute. If no one has questions, I'll just move on. Was there one?
Not seeing any.
Oh, no, okay. Okay. If you don't have questions right now, I'll just keep going to show what native mass spectrum can do for our EMSL projects. And so the two projects in collaboration with Professor Bradley Thibault at the Oregon Health and Science University, and also with Professor Andrew Hellman at Washington State University. And all those were also in collaboration with Professor Vicky Wasaki at Ohio State University, the National Resource for Native Mass Spectrometry Guided Structural Biology. And I got my PhD from Vicky's group. So I'm really trying to bring in the latest technology to help solve the scientific problems at EMSL. And it is very critical because we have those very great technologies that are being developed, and it really helped us to study those heterogeneous protein complexes as shown by the two studies here, which I'll cover in more detail in the next few slides. So the first one is a bacterial biomineralization enzyme, as we call it MINX. And biomineralization is actually a very important process in global biogeochemistry. And you'll find a lot of like minerals in the environment that are mediated by like microbes or some other biological processes. And Professor Tibble was able to identify this bacteria from a marine environment. And he was able to actually recombinantly express this protein, this enzyme. And it's an active enzyme that will oxidize manganese 2 and 3 into manganese 4 oxide. As you can see by the brownish color, that's actually the oxide mineral forming by the enzyme. So what's unusual about this protein is that it does require accessory proteins for stability and function. And this is the SDS page for this protein. So we have the MINX-G subunit, which is homologous to other known oxidases, oxidases in the family. The unique thing about this is it also has two accessory proteins which has no homology to any known structure. And when we had started this project, we were trying to figure out what's the structure of this whole protein complex and what are the potential functions of those unknown accessory proteins. So this complex is about 200 kilodalton, and the ICP results have shown it binds to 10 to 15 copper per complex. The challenge is that when we first started with the native mass spectrometry analysis, we get those the mass determination of this complex, which is about 200 kilodalton as expected. But the heterogeneity of this complex makes it very difficult to even determine the stoichiometry of the subunits, because the two subunits, the smaller subunits, MINX-E and F, they have very similar mass within 1 kilodalton. We can estimate there will be 6 copies total of the two subunits in the complex based on the mass. But it's hard to determine exactly how many there are because of the uncertainty in the mass measurement of the heterogeneity of this complex. The other thing is that we do want to know how many corpus are bound in the subunits, and because of the uncertainty in the mass measurement, it's almost impossible to determine how many corpus are in there. The complex is very hard to crystallize, and it's kind of in the big size range for NMR, and it's kind of on the smallest end for cryo-EM to easily get a structure. So in addition to that, because of the heterogeneity, if we just do a simple mass measurement of the complex, it doesn't give us all the information. So we really have to break it up into smaller pieces and determine the stoichiometry and more of the structure information. So that's basically what we did in this project. And we used both the surface-induced dissociation I showed earlier and also the collision-induced gas collision to dissect this complex. And you can see how different they are. This is because the CID, the gas collision, typically unfolds the protein when you heat it up, and it removes the monomers from the complex. And this gives us relatively limited information on the assembly of the protein. But with surface collision, you can see we get a lot of different fragments, and actually some of them correspond to the release of the intact hexamer of the two smaller subunits from the complex. And this immediately tells us how this complex is assembled. And also, if we look deeper into the released monomers, we can easily see on each of the monomers how many coppers are bound, because now we have a much smaller mass and it's very easy to resolve them. And the two monomers bind different numbers of coppers, and we do have some data suggesting the binding strength is also different. The MINX-ER is a much tighter interaction, and MINX-F is a weaker interaction, which we can actually remove the coppers by adding something like EDTA. And with those data, experimental data, just based on the mass spec data, we can actually start to build models with computational tools. And if you remember the collisional cross-section from the ion mobility measurement, that actually gives us a size measurement of the complex. And this helps us to filter out models that doesn't match to our experiment at all. And this eventually results in a very reasonable model for the complex. What's exciting is that our recent cryo-EM results from Professor Tibel's group in collaboration with LaSalle at Stanford, you can see how the images from cryo-EM closely resembles this model. And you can see there's this hexamer ring on top, and it seems to be binding to the mixed G subunit. So this really shows that with the mass spectrometry experiment, the data acquisition takes only a couple of minutes. You can easily have that preliminary information to start to build models, even without the presence of high order structure. And by looking at the ligand or the meta binding of the protein subunits, you can start to understand what the functions of those proteins might be. And we have an ongoing project with Professor Tibel and also collaborators at the University of Washington, Sasha and Tom Sparrow. We're continuing this work with more activity or functional studies with by looking at the different inhibitors or the different binding of the different small molecules, and also using electron microscopy to look at how the mineral forms mediated by this protein complex. So the second study I'm going to quickly show you is the plant Pesudo-enzyme complex. This protein is involved in vitamin B6 synthesis. You may remember James have mentioned this at the beginning of the conference. So this protein has 3 homologs, and 1.1, 1.2, and 1.3. So 2 of them are active enzymes, but 1.2 in this family is inactive. It has been shown to be essential, so it has some function in there. It's usually correlated to stress of the plant, but we don't really know exactly what the role is if it doesn't have the catalytic function. So the focus of this study is 1.1, 1.2, and 1.3. So 1.2 and 1.3 they have very high sequence identity and homology. And 1.2 has been shown to positively regulate 1.3 activity. So it does help the reaction in some way, but the mechanism of this interaction is unknown. The 1.2 also resists crystallization. If you remember from James' talk, we were able to have a cryo-EM structure of the 1.2, which is very, very homologous, almost identical to the 1.3 crystal structure, except some small differences at the active site. There is a still question on what's the interaction between those 2 proteins. It has been proposed that they interact through a hetero-complex of 2 stacked hexamer rings, one from each of the protein. So with native mass spectrometry, the first thing we did was just to look at the intact mass distribution. So we have the HOMO 1.2, HOMO 1.3 monomers, and then the different co-complex, the hetero-complex at different ratios of the 2 proteins. And this is basically the raw data of this Uncharacterized, and this is after deconvolution using UNIDEC. You can easily see the hetero-complexes, you don't get a very homogeneous distribution. So you have all kinds of different species in there. So this right away tells us that this hypothesis of very defined hetero-complex is probably not the most possible way to explain this interaction. Instead, we're getting this semi-stochastic assembly based on the availability of each protein when you are expressing the protein in the cell. So the next thing we're also trying to really understand if there's any preferred arrangement in the hetero-complex formation. As you remember, James did mention that the cryo-EM data, it's very, very difficult to differentiate the 2 proteins in the hetero-complex. The density map almost just overlap, and there are very small differences at the active site. But when you are looking at the cryo-EM data, because you don't know the position of each of the monomer in the density map, it's hard to define the classes and then start to tease out the different contributions of the 2 monomers. So what we can do, the same kind of concept as I've just shown, is to dissect this hetero-complex using native mass spectrometry. So in the mass spectrometer, we can mass isolate a single stoichiometry of the protein complex and break it apart into smaller pieces. So in this case, the most abundant protein is the hexamer, which means that we're breaking this stacked hexamer rings into individual hexamers. And we also get a lot of other pieces in there as well. And what's interesting is that the hexamer actually has a very preferred stoichiometry. This suggests there is a symmetry in this assembly. It's not completely random. Otherwise, you would imagine the stoichiometry will be more like widely spread out. And to kind of summarize, so the native mass spectrometry, in native mass spectrometry experiment, we can define the variable molecular weight and stoichiometry. And also by breaking up the complexes and mass isolate different species, we can start to understand the symmetry within this assembly. And we have some other data I'm not showing here that suggesting there may be an alternating arrangement of the subunits that will maximize the hetero interactions in the complex. And this is in bioRxiv right now if you're interested in looking into more details. So what this can help us is that we can start to build models to help explain the mechanism of the interaction. So so Irina and James, based on their cryo-EM data, the major differences are there are not many differences in the structure itself, but rather the shape although the shapes are so similar, the surface potential the surface potential are very different between the 1.2 and 1.3. So what Irina did was to build hetero-complex models just by replacing the monomers in the complex. And in this way, we can look at the changes, the effect or the changes of the surface charge when we incorporate a 1.2 into the 1.3 complex. And you can see this is where the substrate binds. And it's once you have the 1.2, you start to change the region, the surface charge in this region. And this may be the reason why there is a supercharging effect that will help promote the release of the substrate, which is actually the negative charge as well. This is like a negative charge, so like the same charge pushes it out to help improve the efficiency of the reaction. And I hope I've shown that native mass spectrometry is very good at dissecting the heterogeneity of protein complexes, and it's also a very fast technique to help us get some structure information, higher order structure information for proteins. And also we can understand some dynamics of the proteins using this technique. And advanced instrumentation is still under active development. So I've shown all the activation techniques. Not all of them are easily accessible right now, but that's probably going to change in the next few years. And you remember this figure from James that so native mass spectrometry is very complementary to other techniques, and it is the biggest advantage as you have seen from the talks today is that it's really good at helping defining heterogeneity of proteins. And if we combine everything, the different capabilities, it really helps us to visualize the proteins in the biological systems to achieve this visual proteomics goal. And with that, this is the end of the talk, and I would be happy to take any questions if you have any.
Thanks, Mowei. There are a few questions. First one from Patricia. How does the licensed software compare to your in-house databases for annotation?
So there's a software, commercial software, and then the other one is not in-house. It's like developed by another faculty member. We haven't done a very sophisticated comparison. I would say it's there are different advantages and disadvantages. So the UNIDAC one is very flexible, and I can go back real quick. It's very flexible, and it's designed for native mass spectrometry experiment because like the developer, Michael Marty, is the native mass spec person. So there are some like more like a very specialized function you can use like for example, membrane protein or nanodisk as special tools for that. For the commercial one, it does use a different algorithm. I've used it mostly for LCMS data because the program is like commercial software, so it's slightly more efficient, like computationally more efficient. So it runs a little bit faster. But I know like UNIDAC is trying to improve at the same time. So it's hard to make a very clear comparison. So if for ease of access, I would just give a unit like a quick try because it's very well designed for native mass spectrometry experiment, and it's free to everyone, and you can just download. For this, it's more designed for like right now, at least right now, for like biopharma applications. So unless you you have like very routine analysis, this is probably like a little more streamlined if you have a like fixed workflow for analysis. I hope that answers your question.
Thank you, that was great.
Next question is from Keith. In addition to in-source desolvation voltages, what other features do the native capable instruments offer to improve large protein ion transmission compared to standard mass spec instruments?
Yeah, so for the transmission, if you remember from Joe's talk this morning, he did show quite a lot of details. I think the biggest thing is like the in-source activation. Basically, like you have like different designs. Like you can trap them and then really kind of heat it up, like by collisions, and that seems to be very effective, especially for membrane protein complexes. The instruments, usually those instruments, you will have to have specialized transmission optics, and you have to change actually the power supply, electrical power supply, because like it's all physics, because you are dealing with much higher mass range, and to effectively trap those, you usually need like a lower frequency RF and different type of like DC voltages, and those are optimized for instruments that are sold for native mass spec. If you're trying to do that on like the regular proteomics, they're not designed like that, and especially for some of them, they also have like software limit on the mass range and everything, can like how you can access them. So so some of them, like the FTSER and the water synapse, they are more like flexible. So and like regarding the mass range, and they have different options, or like for the RF power. So they tend to be more like general. So you can tune it if you are interested. You can just look up the papers that the people publish and what method they use to tune it that way. For UHMR, this is very well designed for native, but some of the other Orbitraps, basically it's a lot of work to try to make it work for native, unless it's already sold for that purpose. So the others, I'm not that familiar with them, so I cannot comment on those if you're trying to get it working on the other instruments.
Okay, this next question is two parts. So first part is it possible to analyze and preserve very weak protein complexes by mass spec? And second part is, could you provide some examples in context of the values of dissociation constants?
Yeah, so those are great questions. For the size, there is not a theoretical limit. So so Albert has shown some very nice examples of very big complexes. And there are also people doing charge detection on very big, like I'm trying to remember what they call the liposome. So it can go very high. It's more like if your mass spec, like if your analyzer optimized to be able to deal with that. So at least from the electrospray perspective, there's not a really theoretical limit. So practically, most of the instrument, you can probably easily go to like mass charge like 10,000, and that probably goes to the range of like megadalton complexes. But if you want to go higher, usually you have to have very specialized tuning or instrumentation. Like the UHMR can go very high, but the others, you usually have to do something special to be able to do that, very large complexes. So the second question regarding dissociation constant, people have been using the native mass spec data to determine the dissociation constant, especially for protein ligand complexes. The caveat for this is if you are trying to quantify the abundance of species across a wide mass range, you have some bias in detection from the instrument because of the different detection optics and everything, or the signal processing. Usually you are trying to do it more like a relative comparison instead of like absolute determination. For protein ligand complexes, there are quite a few papers out there describing like the method. You can probably have some kind of like internal standard to correct for any artifact, but people have already been using that for determining dissociation constant, especially for protein complexes. So I hope that covers your question, and if you have anything specific, just let me know.
We can do one more, I think. I wonder if you know or can control the diameter of the nano ESI tip pulled by the machine.
Yeah. So that's a great question, and it's hard to show all the details, and that's kind of a big variable in the experiment. Like there, so when you are pulling the capillaries, you can set the parameters to pull a different size. It's not that easy because of the control is a little bit limited, so it's a lot of times it's a trial and error. But once you have that condition sorted out, you can check with, especially you can check with like TEM, not TEM, scanning SEM, to check the size of the opening. Once you have that settings, you can reasonably produce like the same size of the capillaries. And usually here we use like on the order of 1 micron up to like 10 micron. It's also depending on what you want to do and what proteins you are looking at. So some of them are very sensitive to, like the spray may be sensitive to the size of your tip. Some of them, if they're very clean, they tend to be okay. They seem to be fine for any size you're using. And I would say that's probably one of the kind of the limiting factor for native mass spec, because that's a kind of a variable, and it kind of varies depending on which library, which protocol you typically follow. But there are also commercial sources you can buy those tips from, and they qualify the size of the tip. It's just more expensive if you do it that way.
Okay, well, we're just over our time now. I think there was maybe 1 or 2 more questions. I'll go ahead and post in Discord for you, Mowei, that you can answer. Okay. I'll put them in the session 4 text channel for everyone to see there.
Great. So thanks you all for joining. So if you have any more questions, just post in Discord, and I'll try to follow up on that.